Hi everybody, this video is here to help you understand the first part of phage hunting. This will take up the first 3 or 4 laboratory sessions. So the goal here is that you'll understand where you're going, why we do what we do, and how it contributes to the ultimate goal, which is to find and then isolate novel bacteriophages from soil samples. So let's start at the beginning. In our case, we are going to be starting with soil samples. So you can see a lovely tree and a nice bush, and just below the surface is that wonderful soil, and it's from there that we're going to take our samples. We're going to bring them into the laboratory. Once we're in the lab, we're going to put those samples in a 50 ml conical tube, pack them with some dirt. We're then going to add some broth to the top. This is enrichment broth, which is 7H9, which is a nutrient-rich solution, not a particularly rich broth, but it's got some nutrients in it, and it's also got carbonicillin, which will inhibit bacterial growth, bacteria other than our host species, and cyclohexamide, which is an antifungal, which will inhibit fungal growth. So we'll add that, and this is so important because this is a soil sample, right? So it's teeming with fungi and bacteria, which is stuff that would complicate matters if we just let anything grow wild in here. So then we're going to shake this tube on a shaker for about 60 minutes, and that's going to mix the dirt with the broth, and hopefully it will shake loose any bacteriophage that is stuck onto the soil particles. So the first session is really just about shaking up your dirt with the enrichment broth to dislodge your phage. And so at the end of that, we've got our tubes. It should be a pretty turbid solution, pretty murky, right? You've just made a tube full of mud water. And so hopefully if you've got phage in there, it's been dislodged from the soil particles. However, now everything is mixed up, and it's going to be hard to isolate that bacteriophage. So what we're going to start to do in order to purify it is centrifuge it. So this symbol here means spin, and so we're going to spin at about 2500 G for 10 minutes. That's going to produce a tube which has all the dirt really tightly compacted on the bottom. We call that a pellet. And we're going to have, hopefully, clear liquid on top, and we call that the supernatant. It's one word, supernatant. Okay? And so if we have phage that can infect our host species, it's going to be in the supernatant. The centrifugation helps a lot. It gets a lot of the dirt and debris out of the solution, but it doesn't remove everything. So we're going to then need to filter this solution. All right, let's change colors for a new day. So we've got our tube with our dirt pellet on the bottom and our supernatant on the top. We're then going to run this through a series of filters. The first one is going to just be filter paper in a funnel, and that's going to drip down into a flask that we set out. And so it's like a 8 or 10 micron pore size, and so this is going to catch all of the visible bits of dirt, bits of twig, anything else that's still in the supernatant floating around. Maybe you've got bark in your sample that's floating on top and didn't pellet out. So that's going to help us with that. Then we're going to take that solution and we're going to filter it with syringe filters. And so basically what we do is we load our sample into a syringe. Right? It doesn't have a needle, but it's a syringe, with a plunger and everything. So we're going to load our sample into the syringe, and we're going to attach a syringe filter to the syringe. And this is just a disc, and the disc has layers of material which are going to filter out successively smaller components of this supernatant until hopefully we're left with nothing but some liquid containing bacteriophage. So you put the sample in here, you push a plunger down, the liquid comes out the other end, and we collect that. And so that collected fluid becomes our, maybe, hopefully, phage sample for testing. right? So we don't actually know, right? We don't actually know if there's bacteriophage in there. Actually, let me clarify. There's almost certainly bacteriophage in here. The question is whether we have bacteriophage that can infect our particular host species, right? Remember, soil is absolutely teeming with bacteria. Wherever there's bacteria, there's bacteriophage. So there's certainly bacteriophage in the tube. We just don't know whether there's bacteriophage that can actually infect our hosts. So that's what we've got to do next, is we've got to test to find out whether there is actually phage there. So we're going to take that nice, it should be pretty much crystal clear, maybe with a slight tinge if there's some tannins in the soil that have leached into the liquid, but it should be not cloudy at all. It should be very transparent. We're going to take that and we're going to do what's called enrichment. And so, you know, we think maybe there's some bacteriophage in here. The number of virions might be very small, and some of those might not be viable anymore. If we're storing this sample for a while, it could degrade in the enrichment broth. So we want to go from that situation where there's very few virions, those would be difficult to detect, to a whole lot of them. And in order to do that, we're going to give it access to its host. And so what we're going to do is, to this tube of filtered supernatant, we're going to add our host, which is Mycobacterium smegmatis. I might just call it M-smeg or even smegs for short, but you should know the name Mycobacterium or Mycolicibacterium smegmatis, as it's now known. So we're going to add that in there. We're going to incubate it, shaking, at 37 degrees Celsius. It's about body temperature. And we're going to do that for anywhere between 1 and 4 days. And over the course of this period of time, what's going to happen is any bacteriophage that's in there that can infect Mycobacterium smegmatis will do so. It'll infect those cells, get its genome into those cells, reproduce, make many copies of itself, break out, infect new cells, rinse, repeat, until you've got a culture that, you know, might have had a small number of virions in the beginning, now has a huge number, and it's greatly enriched for the virions that infect our particular host species. So we call that whole process enrichment. So that whole process is called enrichment, because we are enriching the solution for the particular phage that we're looking for. Now, that's only if there's actually phage in the tube to begin with, right? If we add the host and there's nothing that can exploit that host, we will have gone from 0 to 0. And at this point, we have no idea whether we have anything in there. So how do we actually tell? Well, what we're going to do is we're going to take a Petri dish, and I'll prepare these for you ahead of time, actually. By the time you're watching this, they're probably already done. And I'm going to put a layer of agar in the bottom, a layer of nutrient agar. And this is going to provide a foundation for our next step. So this is a semi-solid, it's pretty firm, firmer than Jell-O, but you could still kind of stick your finger in it if you wanted to. So we're going to take that, and then we're going to add a combination of our host, which again, Mycobacterium smegmatis, that's our smegs. And then we're going to add some top agar. And top agar is like it's the same stuff that we put here, except it's more dilute. So top agar is just a fraction of the concentration of agar as the stuff that we do, what we might call base agar. And so as a result, it's still solid, but it's much more porous. And so things like phages, for example, can diffuse through it more easily. So we're going to combine those. We're then going to layer that down on the top. And so we're going to end up with a layer of top agar above our layer of base agar. Okay, so that's our top agar. And that's our base agar. And now this top agar, because it's the agar plus our hosts, right? If this is our hosts in here, it's going to be riddled with our host bacteria throughout, right? And so a small number of cells at first, but those will reproduce over time when we incubate the plates, and they'll spread and take over the whole agar plate. So let's actually rotate the plate and get a better look at what this looks like. So here's our plate. There we go. Let's see if I can draw a better circle. I'll take it. So here's our plate. We've poured the top agar on it. And now what we're going to do is we're going to take that mystery mystery solution, that filtered solution that we think contains our bacteriophage, or rather we hope that it contains our bacteriophage. We're going to take that and we're going to add a droplet from this tube onto our top agar plate. And so what's going to happen will depend on whether our plate, whether our sample here has bacteriophage in it or not. So if our sample does not have bacteriophage, no phage, then what will happen is our Mycobacterium smegmatis, which is embedded in the top agar, is going to reproduce. It's going to spread through the top agar. It's going to cover the plate. And we're going to see a lawn of growth, right? Lawn of growth. On the other hand, if we actually did have bacteriophage in our sample, then we'll still see a lawn of growth. Yes, phage. Yes, phage. We'll still see a lawn of growth, but in the area where we put the little droplet, right, we're going to drop a little droplet right in the middle of the plate. The place where we put the little droplet, we should have a clear area where the bacteria have been killed, right? So those original bacteria up here have been knocked out, and so they're not going to reproduce, and instead we're going to get a blank area in the middle of the plate. And so that area right here is called a plaque, and this is our evidence of phage activity. Okay, and so if this succeeds, if we get a plaque where we put the droplet, that means we have bacteriophage that can prey on, use as a host, Mycobacterium smegmatis, and we're ready to go on to the next step. I don't know how many of our samples are actually going to yield phage in this way. It could be just a few, it could be a lot, but however many we get, we're going to total those all up. We'll divide them among all of the participating groups, and we'll all move forward together. And that's actually a good thing to not have everybody succeed, because it means we can use, we can do the rest of the testing in duplicate or triplicate, and that'll make our results more reliable. So I hope that this is helpful. This is the first part. Once we find that we have phage or not, there's a number of additional steps that we're going to take to purify it, confirm that we have just one phage in a sample rather than multiple different ones, and then amplify it, like make lots and lots of them, do a DNA extraction that we can then use for sequencing, and also prepare samples for electron microscopy. So that's later. We'll get to that in another video, but for now I hope this is helpful. Let me just do a quick review again. We start with a soil sample. We shake that with some broth to detach phage from the soil particles. We then spin down that suspension of dirt and phage. We get a pellet of dirt at the bottom and a supernatant, which maybe contains our hopefully bacteriophage. We then take that supernatant and we filter out any remaining dirt, silt, random soil bacteria using filter paper and then syringe filters, perhaps a series of syringe filters depending on how dirty it is. We then take that filtered supernatant and we add our host bacteria, M. smegmatis. We shake that in the incubator for a couple of days, allowing the bacteria to reproduce and for the phage to reproduce inside the bacteria, amplifying their numbers. Then we make a top agar plate that contains our host. We add our filtered maybe phage solution to the center of that plate. If there's no phage in that solution that can infect our host, we'll end up with just a lawn of growth. If there is phage in there that can infect our host, we'll end up with a blank spot in the middle of the plate, and that's our evidence of phage. All right, so I hope this is helpful, gives you some perspective on where we're going for the first part of this investigation, and we'll see you in the lab. Bye-bye.